Welcome to Kognito Key. Have you ever wondered what it takes to make the absolute invisible, not just visible, but completely radiant? Imagine peering into the microscopic world, right? And instead of a blurry gray blob, you're seeing these vibrant, glowing, neon structures popping against a pitch-black background. It's honestly like looking at a tiny galaxy. Today in this explainer, we're unlocking the secrets of the brilliant and frankly fascinating world of fluorescence microscopy.
The sheer visual poetry of this technique is just breathtaking. I mean, in traditional microscopy, looking at a cell can kind of feel a bit like staring through a foggy window. But fluorescence microscopy, it creates this definitive, striking contrast. By using highly specific reactive chemical stains, the unstained portions of a specimen simply vanish. They fade into this jet black background. And the targeted cells, they glow brilliantly. It's exactly like turning off all the lights in a dark room and finding exactly what you need with a glowing neon highlighter.
Part 1. The Science of Fluorescence.
So to really understand this, we have to look at a physical mechanism first studied by a scientist named George G. Stokes way back in 1852. He coined a term called the Stokes shift. You see, fluorescence happens when chemical compounds, which we call fluorophores, absorb light energy and then emit it right back out. But here's the catch. They don't just act like a standard mirror reflecting light, they physically transform it. The difference between that absorption phase, known as excitation, and the release phase, known as emission, well, that's your Stokes shift.
And the timeline for this transformation? It's almost impossible for the human brain to fully comprehend. When scientists say there's a short delay between excitation and emission, we aren't talking about seconds. We aren't even talking about milliseconds. This delay takes mere nanoseconds. To your eye, it's practically instantaneous for sure, but in the realm of physics, it's a definitive, measurable gap where those high-energy photons drop back down to their ground state.
Let's actually break down those two distinct phases. On one side, we have the excitation phase. Here, our fluorophores absorb short-wavelength, high-energy photons, typically ultraviolet or an intense blue light. This energizes or, excites the molecules. Then comes the emission phase. As those excited molecules finally relax, they release that trapped energy as long-wavelength, lower-energy visible light. Essentially, you put invisible, high-intensity short waves in, and you get these beautiful, visible, glowing long waves out.
Part 2. The path of light. Before our light can even start its journey through the microscope, we need two things: a powerful source and a specific target. For the light, these microscopes use intense beams. We're talking xenon or mercury vapor lamps, high-power LEDs, or even precision lasers. Down on the specimen slide, we have our targets tagged with those fluorophores we talked about. Dyes with names like Hoest and Phalodin, they act like tiny fluorescent homing beacons. They're honestly incredible because they specifically hunt down and attach only to particular biomolecules. Think of the DNA inside a nucleus or the actin fibers that give a cell its physical shape.
Now this is where the mechanical magic actually happens. First, our incredibly bright light hits the excitation filter. Think of this like a bouncer at a club, only letting the correct, short-wavelength light pass through. Second, this light strikes what's called a dichroic mirror. This is a brilliant beam splitter sitting at exactly a 45-degree angle. It bounces those short wavelengths straight down onto the specimen. Third, the light hits our fluorophores, exciting them so they instantly emit that longer wavelength green light. And finally, step 4, this new green light travels back up, passes smoothly straight through that same 45-degree mirror, and goes through an emission filter, delivering only a pure, glowing image right to the detector.
Part 3. Advantages and drawbacks.
Okay, so the advantages of this setup are absolutely staggering. The sensitivity is so remarkably high, we can actually detect as few as 50 molecules per cubic micrometer. That's a mind-boggling level of detail. And because we can use differently colored fluorescent dyes all at the very same time, researchers aren't just looking at static dead structures anymore. No way. They can watch dynamic living cell behaviors unfold right before their eyes in real time in full radiant color. It's a real game changer for biology.
However, we do have to be objective about the physical limitations here. First off, you only see what you successfully tag. Everything else in that microscopic environment is entirely invisible to you. Second is a pretty frustrating issue called photobleaching. Over time, that constant excitation causes the reactive dyes to permanently lose their emission intensity. The glow literally fades into darkness. And finally, there's the phototoxic effect. Blasting living cells with high-energy photons can actually harm or, unfortunately, even kill the very microscopic life we are trying so hard to study.
But
Even with those physical limitations, fluorescence microscopy remains one of the most indispensable keys we have to unlock the deepest secrets of biology. Which leaves us with a pretty fascinating question. If we can light up the invisible inner workings of a single cell today, what hidden universes will we illuminate tomorrow? Thank you so much for joining me on Kognito Key. If you want to keep exploring these mysteries with us, go ahead and hit that subscribe button, like, and share this explainer with your fellow curious minds. See you next time.